Well, welcome to the Get Fit with Jodelle podcast. I am, as usual, Jodelle, and I'm very excited to have my special guest with me today, who I have been listening to and have read numerous papers on, and I just really want to inform you guys of what this individual is putting out there and why it's so important that I share this today. So my guest is here to share his recent work on reporting bias in the COVID-19 vaccine specifically. And I brought him on to help us understand the myths around understanding how this COVID virus and the vaccine affects us. Again, this podcast is for, just like the one I did previous to this, it's for educational purposes only and should not be construed as medical advice. A vaccine is a very personal choice that each individual should make for themselves, but we do want to inform you guys of the truth surrounding it so that you're able to make solid, informed decisions. So have you heard of relative risk reduction or absolute risk reduction? Did you know the difference? There is. And did you know the human body has a virome similar to a gut microbiome, which plays a role in maintaining overall health in the body? We're going to discuss that. Plus, sodium chloride is the solution the vaccine comes in. Seems to be harmless, but this PhD holding top researcher from the University of Waterloo, Dr. Ronald Brown, that's who's with me today, has some interesting things to say about it. As well as he recently has stated that the COVID-19 fatality rate is the worst miscalculation in the history of humanity. Dr. Brown, I'm so interested in hearing you inform my listeners to the behind the scenes info that no one else is really talking about. So fill us in. Why, as a research, were you led to do so much work on this virus?
Well, first of all, thanks, Jodelle, for inviting me. I'm really happy to have a chat with you. And just listening to your introduction, that is the most impressive introduction I have yet had from any podcast or broadcast or whatever. Thank you very much.
Well, thank you.
And the difference is obvious. You've been reading and listening to what I've been saying.
Absolutely.
So good for you. And I don't know how much more I can even add on top of that, but what we can do is, just go through all of these things for your audience to make sure that, they're exposed to it. And let's get a discussion going. Let's have a debate. If people want to challenge me, that's okay.
Yeah.
That's what science is supposed to be about. When people say, oh, you should listen to me because it's science, you should reply, oh, that's nice. Which side of the scientific debate are you on?
Mm-hmm.
Because there's always two sides to the science story, right? So don't let anybody ever tell you that you should listen to them because it's science. Right. There's always two sides to the story. And that's what we call debate. But of course, we have censorship. We don't have debate anymore. We're under these emergency orders. I'm in Canada, Ontario, Canada, and we're still under lockdown here. And what is it, May 29th, 2021. And we're probably going right into June. And where did this start? Let's talk about that.
Yeah.
Because I've done some research on how we got into this mess in the first place. It all goes back to Wuhan in 2019. There were 4 cases of pneumonia in Wuhan. A lot of people don't understand that the coronavirus was first identified in 4 pneumonia cases. But the problem was, when the researchers looked for the pathogen that's usually associated with lower respiratory tract infections like pneumonia, they couldn't find the usual pathogens. So they tried out this newer technology that we now have, genomic sequencing, and they discovered a whole new genome sequence for the pathogen that was associated with the pneumonia in these 4 people. And that was named eventually SARS-CoV-2. It was like SARS-CoV, the original SARS virus that was an epidemic. It was actually a pandemic, but it was an epidemic in China back in 2003. It was very close to that, but they found a little genomic difference. So therefore that entitled it to be called a new virus. Now, I have a problem with that.
Okay.
It's a new genomic sequence because we've only been doing genomic sequencing for a few years. It only started like 15 years ago. So there are trillions of life forms that need to, be analyzed with the genomic sequence. So we're discovering new ones every day. Does that mean that every time we discover a new genomic sequence, that's a new life form? No, it might be, but probably not. See, that's the difference between association and causation, right? Just because we have a new genome sequence that's associated, with a pathogen, doesn't mean that that's a new pathogen, right? Especially considering that this technology is so new. I came across an article in 2017. It was called Genomic Epidemiology, a peer-reviewed article. And it told us about how genomic sequencing is going to change all of infection control worldwide and how great it is. And 99% of that article was just describing the genomic sequencing. And then they got to the epidemiology part. And they said, oh, yeah, and by the way, all of this stuff needs to be vetted by epidemiologists to make sure that that sequence of that organism is actually, spreading, is a severe risk or not. We can't just jump to conclusions. Mm-hmm. And then I thought, but that's exactly what we're doing. We're so enamored with all this new technology, these new genomic sequences of this virus, this new virus, and then all the variants, and all that information is going directly. We call this being translated from the science, the technological science, over to the people making these decisions, the public health people, right? The politicians. What happened to the epidemiologists that were supposed to vet all this stuff? Just like that article said.
Yeah.
They're off in the background going, wait. We need longer surveys. We need serial surveys to see how widespread this exposure is within the population. We need control groups. Just because you're going out and testing groups of people and you're finding this, it doesn't mean that it's spreading within the groups. Did you bother to do a control group with another group of people to compare? Let's say you have one group and they're close together. So you say, oh, we got to target those people for testing, right? And sure enough, you find the prevalence of the virus, the infection there. Did you bother to compare that group with another group with the same type of people, but who perhaps are not as close together, to see if there's any difference in the prevalence? No, I never hear that, ever. Maybe some people are doing it, but basically, that's exactly what I mean by we're leaving out the vetting, the epidemiological vetting, because that's what an epidemiologist would do, for example. You do these case control studies. You take cases and you match them up with controls, right, to see if there's the same amount of incidence or prevalence. Mm-hmm. And that's when you can come to a conclusion. But we're not doing that. It takes time. So as a result of this, we have this Chicken Little policy, right? The sky is falling. We discovered a new pathogen we never saw before. It's spreading and killing us. Hang on. Yeah. Maybe it is. And I'm not ruling that out either. Mm-hmm. But before you come to a conclusion like that, you have to have a lot more evidence. Mm-hmm. And that's the problem. So getting back to Wuhan, I talk a lot, as you can tell. Just jump in anytime you want to interrupt.
That's what these are about, man.
And I go off on tangents, but pull me back in again. So getting back to Wuhan, so there are those 4 people. Oh, we never saw this pathogen before. It's a new pathogen. We better warn the world, right? How do you know it's a new pathogen? How do you know this pathogen hasn't been around as long as we have? Mm-hmm. What's new is the genome sequencing technology. And there are like, well, I guess it's approaching 500,000 genomic sequences of the coronavirus, right? So what are we going to do? We're going to have, the sky is falling policies every time we discover a new virus. And then this is for the species coronavirus. What if we come up with some other type of pathogen? And we start, and that's associated with some kind of respiratory illness. And then we discover a new sequence, genomic sequence. We're starting all over again, right? We can't afford to do this anymore. We're losing too much. So we have to get smart about this. Mm-hmm. We got to start using science the way it was intended to be used. And it's interesting. And I'm still going to get back to those 4 people in Wuhan. Don't worry. Okay. But when I talk to people, I can tell right away whether they have any background training in what we call research methods or not. Most people don't. I have a lot of extensive training in research methods, right? And so you're taught to think critically, to think a certain way when you look at evidence in the way that normal people, and there's nothing wrong with the way normal people are. They're normal, but they're not trained
right
to think that in that kind of critical way. So I see all these common errors, like, oh, those 2 things went together, therefore that caused that. No, you can't say that. But people aren't taught to challenge that way of thinking. So we have a long way to go to bring the public to, we have what I would call an illiterate public, the public illiteracy in basic research methods.
Yeah.
I mean, we have reading, writing, and arithmetic, right? We need research methods also. Can you imagine if we lived in a society where people couldn't read, write, or do arithmetic? And then they were dependent upon the so-called experts to do it for them, except those experts weren't much better at doing it either. I mean,
I feel like we're in that.
Yeah, that's exactly what we have, right? So we've gotten away with, tolerating our illiteracy.
Yeah.
Yes, we can't do that anymore. If there's one lesson we learned after going through all this, is that we have to be educated about all this type of information. We have to learn to think critically. Now, getting back to the 4 Wuhan people again, so those technicians and the people that, the virologists who were investigating this, they decided to call this disease novel coronavirus pneumonia. Okay? They're still calling it pneumonia. The World Health Organization doesn't call it novel coronavirus pneumonia. They call it COVID-19. By the way, the World Health Organization doesn't call the virus SARS-CoV-2. SARS stands for severe acute respiratory syndrome, right? Coronavirus 2. They call it, the World Health Organization calls it novel coronavirus 2019. They named the virus to be identical with the disease. That's a nomenclature error in medical nomenclature. You're not supposed to do that. By the way, this is coming out in my latest article, my 3rd article on.
You were getting a sneak peek.
Yeah, well, just talk about this stuff. I have basically 4 articles, right? 2 are already peer reviewed and out, and they're killing the views in the journals that they're in, right? Yeah. And so the other 2 are under peer review right now, but I can talk about the evidence at least. And by the way, I'm one of the few talking heads that can talk about their research. I'm not just expressing opinions, right? Yes. Peer-reviewed research.
Yes.
So I've spent almost a year and a half now, however long it's been, just working on this. I transferred all of my investigations from cancer and heart disease and other diseases that I was investigating. And I just worked on this. Why? Because my university is shut down, basically. I mean, yeah, we can have these little Zoom meetings, but nothing gets done that way. Right. I'm sorry to point that out to people, but I can't tell you how important it is for me to walk into my supervisor's office and sit down and just start chatting, kind of like what I'm doing right now with you, and sharing. That's where the real sharing of the ideas happen. And we could do it over Zoom, but you have to schedule a Zoom meeting and stuff. It's not the same as just having that, intimate, instantaneous contact. It's just not the same. Things just don't get done as well, right? So anyway, that's why I'm doing all of this research.
Well, I guess what I want to bring up is I love that you said critical thinking, because I feel like that's what's missing. And especially like I was taught as a nutritionist in my training how to read a research paper, but a lot of people aren't. So they just take the abstract or the results and they go, oh, okay, that's what that means. Well, there's so much more to it than that. And so that's why I wanted to have you on. And I want to encourage people, don't just follow what's said on Facebook or what your neighbor's doing or what even your, Channel 3 News says. You have to start doing critical thinking and go, why are these things so? What did we do in an instance like this when we had a viral eruption, at some other time in our life? Was it ever this way? Should I start asking more questions? So that's why I wanted to have you start talking and start to bring up the right situations for them to start asking why with, such as relative versus absolute risk. Let's go there too.
Yeah. Before we get there, you brought up a couple of things.
Yeah.
Facebook. Now, my article on the vaccines was published in Medicina or Medicina, depends how you pronounce it. Yeah. And it's the number 1 downloaded, it's almost the number 1 downloaded article in the history of that journal. And it's definitely the most viewed article in the history of that journal.
Cool.
And so there's Outmetric. I think that's the name of the online surveying company that keeps track of your exposure from that article. And according to their statistics, I'm approaching about 3,000 Twitters, right? Twitter messages, but I only have 2 Facebook messages.
Weird.
2, right? So you know what that means. I'm being censored, right? because it has something to do with vaccines that doesn't exactly follow the narrative, right? The conventional narrative. And so now I'm just going by that evidence, but the evidence shows clearly that how come the Facebook views aren't anywhere near the Twitter views, right? So there it is, and it's just come out over the past few days that Facebook has had a policy to censor mention anything having to do with vaccine information that's contrary to the established narrative, right? Another thing I wanted to mention before we get into the vaccine stuff was you said, well, how did things used to be before, when a new pathogen emerged? We never shut things down.
No.
What did we do then? Why is it different now? And that's beautiful. That's absolutely correct. Take like Ebola, for example, which is a pretty
exactly. That's what I was thinking.
Right?
Yeah, that's what
so how come we're like we all didn't just pack up and move to Mars or something to get away from Ebola?
Yeah, it's much more deadly.
Because we still had a reasonable system in place. We had the warnings, and then the epidemiologists took over and started to look into it, to see how widespread is it, how deadly is it, how much of a threat is it to us, right? And then eventually they reported back to the public health officials, who reported back to the politicians that not a problem, don't worry. Some people, yeah, in some areas, yes. But for us, no. That's the normal way it should be done. You see that? You have the technicians, like, for example, who discover the pathogen, then being vetted by the epidemiologists, right? Those longer term studies, and then passed off on to the public health policymakers, right?
Yeah.
That's the missing link that we have now. It's because the technology has become so rapid, and expanding so much. Like if you want to make a lot of money, get into genomic sequencing and start a genomic sequencing company, because that's how rapidly it's push the epidemiologists out of the way. And so I think I made that point already. Do you know, for an example, there's a national United States sero survey that was started more than a year ago, for the whole population of the United States. It was actually started by Dr. Fauci's institute, the National Institute of Allergy and Infectious Diseases. And he never even talks about this, right? And it's going to continue and be completed next March 31st. So it's a 2-year sero survey study to see exactly how widespread is this exposure to the coronavirus and how deadly is it. In other words, what is the infection fatality rate? Not just the case fatality rate. Now, this I talked about in my first article. There's a distinction between the case fatality rate and the infection fatality rate. Of course, now the World Health Organization just destroyed that. They conflated it. So now if you have an infection, you have the disease, you're a case. Even if you're not a case, even if you don't have any symptoms, right? How do you define a case? You have to be sick. Literally sick. You have to have clinical symptoms. Not anymore, according to the World Health Organization. As long as you've got that laboratory confirmation, that's it. You're a case. So obviously that makes the case fatality rates go much higher than they should. It's the infection fatality rates that you need to look. All cases are infections, but not all infections are cases. So if you're an infection, but you're not sick or anything, you're in a different group of people, right? And that's the group of people that's being examined in the sero survey that's going on in the United States for 2 years, right? And that's the kind of information that will tell us once and for all how deadly and how severe and how widespread, this virus is. Until we have that, everything we're doing, all of our policies are based upon anecdotal evidence, wild speculation, and fear mongering.
Yes.
And control. You name it. You can just pile anything onto that assessment. So that's one of the problems. Do you want to jump in or?
Yeah, because what you're saying is this is such a good conversation, because I want people to think critically, and that's what you're doing, is you're getting the wheels spinning of people to think critically and go, okay, yeah, I've never thought about it that way. And not, like you said earlier in the podcast, not that this isn't something that should be a concern, but yet to what degree? That's what we don't know. And what I love about the relative versus absolute risk is it kind of breaks it down a little bit. So do you want to go into that?
Just before we do that,
okay.
I got to tell you another thing about this new article that's hopefully coming out, and how. Let's go back to China again. Okay, So what China did in February of 2020, so when things were just starting to boil over, all right, they started experimenting with their case definition. So how do you define a case of coronavirus? Notice I'm saying case, not infection, because a case now includes an infection. It also includes clinical symptoms, and it can also include epidemiological evidence, like who have you been in contact with, right? Contact tracing and things like that. So they were experimenting with that. They decided that they would broaden the case definition in February 12th, and they would allow anybody who has pneumonia, just clinical symptoms of pneumonia, to come in, to see if they could be considered part of a novel coronavirus case. Well, their cases exploded sevenfold in one day. And you can look on this on worldometers or any other type of graphic. On February 12th, it just, cases shoot up like a skyrocket. So I guess the Chinese government said, no, we can't have that. So they changed the case definition to eliminate that clinical evidence. Cases came right back down again the next day. You can see this, it's recorded right there. And they must have thought, and I'm speculating a bit, but based upon what's happened next, they must have thought, let's keep on narrowing this case definition, So I said, okay, so in order to be a case, you have to have pneumonia. If you don't have pneumonia, you're not a case of coronavirus. Well, there's a big difference right there compared to what we do. We do just the opposite. Anybody who has the infection is a case, right? Remember we talked about that.
Oh, yeah.
China does the exact opposite, because they're not following what the World Health Organization is doing. The World Health Organization is following China, as I'll explain in a second. So they narrowed their case definition. You have to have pneumonia to be a case, but they went even farther. And this is their genius, right? They said, not only do you have to have pneumonia, but there can't be any other pathogen other than the coronavirus present when we examine you. After all, this is novel coronavirus pneumonia. If there's another pathogen, then you're not novel coronavirus pneumonia. Yeah. Even if the coronavirus is there. And guess what? Coronavirus is usually there in over 86% of other cases, other infections. In other words, 86% of the time, coronavirus will be co-infected with some other pathogen. So China eliminated that. They said, no, that doesn't count anymore as a case. So now they eliminated another 86% of the cases. There's nothing left. You know how many people died of coronavirus in China in the past 12 months? Where are we now? We're in March. We're in May 29th, 2021. Okay, for a year ago, how many people have died from the coronavirus in China? Take a guess.
Well, if you narrow it down, if they're counting that low of statistics, I'm going to think like 500.
That's a pretty good guess. Okay. You're on the right track. Okay. How about 2?
Oh, gosh.
Not 200. 2 people.
Wow.
You want proof? Go to Worldometers, and check it. And you'll see under cases in China, China, 5th country or something in their index, and then there's a flat line for like over a year. Just literally flat. You have to go back a year to count up 2 deaths. Okay. Now, why is that? Yeah. Are they so wonderful doing things there? It's so healthy, we should all move there? No, it's because their case definition has eliminated all of these cases. And if you eliminate the cases, then you eliminate the deaths, right? Yeah. That's it. They're still dying of pneumonia, right? That hasn't gone down.
No, that's not going to stop. That's normal. Yeah.
Yeah. Right. So, but it's all now the case definition changed. Now, the World Health Organization saw this. They didn't realize that China had changed their case definitions. Right. There's this guy named Bruce Aylward. He's one of the directors of the World Health Organization. He was in charge of working with China back in February of 2020. And he saw this tremendous drop, over 80% in February. Remember when first it went up, and then it came way back down, and then it just kept on going down. And he looked at that and he said, wow, what are they doing here? They're locking everything down, because that's what they did while all this was going. While they were changing their case definition, they locked everybody down, right? And they did all the contact tracing, all the things that we know don't work. And who said that anyway? The World Health Organization, by the way. All those what they call non-pharmaceutical interventions, those mitigation measures, weak evidence. So I'm not going to say they don't work completely. I should just say there's weak evidence that they work at all, right?
Yeah.
So China was doing that, but mainly what they were doing were their draconian lockdowns. Nobody in the world had ever done, at least in modern times, had ever done lockdowns like that before.
Right.
It would take a communist country with an iron fist to, rule over people's rights to get away with that. Sound familiar? Mm-hmm. That's what we have now. And you know why? Because Bruce Aylward saw that in China back in February 2020, didn't know that it had anything to do with the case definition. And he said, wow, everybody has to do what China's doing. All the countries adopt these lockdowns that China is doing because that's what worked. And there's the proof. See, 80% drop, right?
Yes. So what I'm hearing is...
And that's what we did.
Yeah. What I'm hearing is it's not so much that the cases have exploded. It's technology changed with the genomics, and then the case definition changed.
Got it.
And then
You're
also,
right. I like it.
And then also the one individual noticed an issue of like, let's keep people under control, and they formed a solution to how are we going to maintain this virus.
And that's it. And the World Health Organization, I don't know if they know the truth by now. They will if they listen to me. I'm not sure, but they never retracted that. We're still stuck in it. And just because we're coming into the summer now and things are calming down, right? And we can talk about that too. But that doesn't mean that next season things aren't going to start up all over again. And now the cat's out of the bag. The toothpaste is out of the tube, right? The genie's out of the bottle.
Yeah.
Once it's out, we're going to go back to that over and over again. Why not? Look how well it worked, right?
Yeah.
Well, when we lock down things, cases drop. There's that critical think or lack of critical thinking again.
Right. Yeah.
You can't say that. You don't know. How do you know it's just not seasonal influenza or pnemonia?
Yeah.
During
or lack of vitamin D.
Or lack of, that's kind of related to it because the climate change, is related to the seasonality of respiratory infections. Mm-hmm. We've known that for years.
Absolutely.
At the end of winter, especially in the northern latitudes, beginning of spring, end of winter, there's an epidemic, usually a local epidemic, of lower respiratory infections, pneumonia. The ICU's filled up to capacity.
For sure.
Every year. There's nothing new about this. Mm-hmm. They get to the point of overcapacity. Because think about it. A hospital can't build up its capacity just to accommodate, one brief part of the season every year, and then have all that capacity just sit there around doing nothing for the rest of the year. It's way too expensive to do that. Hospitals are purposely budgeted to only arrive at their maximum capacity, once every year, and that's it. They can't go beyond that, right? So every year we have what we call hallway healthcare. People left out in the hallways, because there's no place to put them, right? Mm-hmm. That has nothing to do with the coronavirus. Why? How do I know that? Because we've been having that ever since, years before the coronavirus ever happened, okay? So there's that correlation does not equal causation problem again. People are associating that with, the coronavirus is causing all this. Okay, so let's get to the vaccines.
Yeah. So relative versus absolute. I'm waiting for this one because I want everybody to understand this.
You know what? Oh, I got something I want to share with you.
Okay.
Because what I'm going to tell you, already, but your audience may not know. So we'll go through it. But at the same time, what good is it if you know it and I know it, but Dr. Fauci doesn't know it? Or does Dr. Fauci really know it? And he's just not telling us. There was a clue. There was a YouTube video where Dr. Fauci is talking in the White House. It looks like it's the White House. He's sitting next to Deborah Birx. So it must have been when he was on the committee, the advisory committee then. And he's reminiscing about the HIV trials and the drugs they used back in the 80s for HIV. And he was talking about the outcomes and how the drug for, to treat HIV was slightly more effective than the placebo. So the drug reduced the mortality. The mortality for the drug was like 8%, and the placebo was like 11%.
Okay.
So there's a difference of 3%, right? That's called the absolute risk reduction. Okay. How much is the difference between the treatment group and the placebo group? And he was saying it was only 11, it was only 11% in the placebo group and 8% in the treatment group, right? And then he said it was not yet statistically significant. And then he said, but it needs to be further analyzed. I told you you're smart. Exactly what I said. What is he talking about? I'll tell you what he's talking about. He's talking about we need to statistically manipulate it into the relative risk reduction, which then would, in that case, would have vamped it up to over 20% or something, right? Which was enough to declare a success in the trial, right? But so he knew it was all a question of how you analyzed it, right? It's not like he said, well, we need to collect more data. In fact, he said the opposite. We have all the data we need. But all we have to do is, a little more statistical analysis, right? Why doesn't somebody ask this guy this? What were you talking about? Were you talking about the relative risk reduction? Okay. So I suspect he knows all about this and he's just keeping quiet. So what we need to do is figure out how can we get our Congress representatives who meet with this guy apparently on a regular basis, because every week, they're yelling at Fauci and Fauci's yelling back at them in Congress. So let's arm these representatives with some questions. So I wrote some questions down. Vaccine questions for Dr. Anthony S. Fauci, right? And I want to post this. I'm working with trialsitenews.com right now. And so I'll be able to post this on their website. My interview I did with them on YouTube is it's approaching 200,000 views now. So it's going pretty well. And I predict if it keeps going at that rate, by the fall, it'll be over 1 million views. So they kind of like me there, right? So I'm writing this stuff and I can kind of get this posted, but I want to go over this with you and see what you think about this, right? Because, so the relative risk, so you see, if you take the absolute risk reduction, which I already explained to you was simply the difference, the mathematical difference between the percentages of the two groups in a randomized trial, right? Whatever the treatment group is and whatever the placebo group is, okay? And so even if it's small, what you do to convert that to the relative risk is you divide it by the risk in the placebo group. So why would you do that? But kind of intuitively, what it kind of represents is you have the reduction in the risk of the disease according to the treatment. That's the absolute risk.
Right.
How does that compare? How is that relative to? How is it divided by people who didn't get the treatment? In other words, the rate in the placebo group. That's the idea of how we're coming up with the relative risk. But there's a sneaky little mathematical property here. Whenever you divide a percentage by another percentage or any number by a percentage, usually when you divide a number by a number, you get a smaller number, right? If you divide a number by a percentage, you get a larger number, right? That's the mathematical trick behind this. So that takes that absolute risk reduction and makes it a larger relative risk reduction. So in the case of the vaccines, the messenger RNA vaccines, the absolute risk reduction of Pfizer is 0.7%. It's 0.7%. When you actually subtract the rate of the infection of the treatment group from the placebo group, it's 0.7%. It's not even 1%. Moderna is 1.1%.
Okay.
Right. So then if you divide that absolute risk reduction by the risk in the placebo groups, it blows up to 95.1% relative risk reduction in Pfizer and 94.1% in Moderna. That's it. Okay, and that relative risk reduction is called the vaccine efficacy. If you look up vaccine efficacy, it means the relative risk reduction. They're identical. That's the same. Okay, and this has been going on for decades. This is nothing new. No. In fact, in one of my epidemiology courses in my university, there in my lecture notes, it mentions this. It mentions that, you have to be careful in interpreting the relative risk reduction. It's not the same as the absolute risk reduction. The absolute risk reduction gives you the information you need to determine how effective clinically and from a public health point of view the treatment is. The relative risk reduction misleads you. It doesn't have anything to do with that. So why do we have the relative risk reduction? It's used by researchers just to quickly do a quick comparison between different treatments. That's it. It's kind of like the body mass index. You know the body mass index?
Yeah.
Right? And how there's problems with that because
we don't hardly even use it.
And we don't hardly even use it. But the thing is, it's not supposed to be used for individuals. It's supposed to be used to generalize entire populations.
Yes.
The same idea with the relative risk. It's not really intended to be used for individual effectiveness for you or somebody else clinically or from a public health point of view. It's used to compare large segments or large trials with each other. That's it.
Yeah.
They don't even use that anymore. Now they use what's called the number needed to vaccinate, which is basically the absolute risk reduction turned upside down, the reciprocal. That's all that is, right? So if you want to do a comparison, you can use that.
Yeah.
But no, we're still using the relative risk reduction. Even the journals, all the journals say don't, not all of them, but they say don't use this. In fact, my article was just cited in the Lancet last month, bringing up this issue once again.
Yeah.
So why is it different now? You see, a year ago last March, I still think we're in March, we're in May. When I first heard the discussion that, well, we need a vaccine for this, I thought, for the coronavirus, I thought, okay, this will be a good time to bring out the absolute risk reduction issue, right? Why? Because everybody's going to be paying attention. They're going to be laser focused on this. And so we can see this all happen in real time. Perfect. And so I got the manuscript ready. All I had to do was wait for the final results to be published in the New England Journal of Medicine, which finally came out by the end of the year, December 30th, 2020. The Moderna final data from the clinical trials was published in the New England Journal of Medicine. I had everything I needed. So I just tweaked the numbers in my manuscript, sent it off right away to the journals. And I finally got it published in Medicina, right? And boom, just exploded because people want to know this now. Now, if there was no coronavirus pandemic and I wrote an article like this, would we be even having a discussion now? Of course not, because nobody would pay attention. So you see, that's the difference. It's all a question of timing. So I'm not saying anything new here. This is all well known, but it's about time that the public got educated about this, because if you're making these decisions without full informed consent, that's in violation of your legal and your ethical obligations, your legal rights and ethical obligations.
Yeah.
It's a big problem. It's misleading, and I'm gonna just look at my article here. It's misleading, and it's just not useful for clinical and public health purposes, right? There are better things that we can use, the absolute risk reduction.
So with the relative side of things, then you have the information that's out there about how the vaccine is so protective, and like, this is the risk reduction. You don't have to worry about it anymore. You got your vaccine, you're good. And we're not saying that they can't do a vaccine, but I think they should understand what the vaccine does not do. And that's where I want to go next with you, because you have a lot of information about what it does not. Because I walked into a library the other day that had just opened, and the librarians said, "Oh, you don't have to wear your mask because we're all vaccinated here. We're all good." And I wanted to say, are you though? Like, there's some truths that I think you've been told that maybe aren't true after you get your vaccine, such as it doesn't actually protect against COVID-19. It only lessens the symptoms, correct?
Correct. You're so smart. I mean that sincerely. I'm not just joking around with you.
Yeah. Okay.
You'll have to look at the endpoints of the clinical trial and the way it was designed, right? So that it was designed to test for a positive coronavirus test, PCR test, and at least one clinical symptom. In other words, what we would call a mild to moderate infection. That's it.
Right.
It was not designed to test asymptomatic infection transmission. Right. It was not designed to test severe infections, hospitalizations, or deaths. To do that, you would have to have different types of groups of people, right? The groups of people that were used in the clinical trial were all healthy people. They didn't have underlying health conditions, right? So if you want to test for how effective it is in a clinical trial, to prevent hospitalizations and deaths, you need a whole different type of group of people. You need people with underlying health conditions. So you see how you can make observations about, well, the people in our trial, of course, they were healthy, so obviously they weren't dying. So it must prevent deaths. No. You're wrong group of people. You need a whole other group of people to study to make that. You see the point?
Yes. Mm-hmm.
Or asymptomatic transmission. That's where the control groups and stuff come in. That's where the exposure from the antibodies, the serotesting comes in to do that kind of test, right?
Right.
So that I hope that answers your question. That's all it does, but that's not what people are being told. They say, "Oh, this will protect you. It'll make you rich. It'll make you beautiful. It'll do everything you want it to do."
And it doesn't prevent the spread. So like you can still spread the virus. And
Yes.
so they're thinking, I'm good. I've got my vaccine. I'm great. I don't have to worry about getting it or transmitting it.
Well,
so I just think there's a lot of false information out there.
When you bring up, it doesn't prevent the spread. I know what you're trying to say, and I agree with you, right? Okay. It doesn't prevent it. However, my fourth article, I talk about virology in general and how this whole idea, all the premises that virology is based on are pretty much wrong. All.
Yeah.
And I don't know if you've been exposed to any of that information yet, because I haven't really talked as much about it. But the idea that viruses can spread is just not supported by the evidence. It's supported by anecdotal evidence. There's no causative evidence. Mm-hmm. The idea that a virus can replicate, again, the proof of concept is so flawed, it's silly. It reminds me of a magic trick. I used to do magic tricks when I was a kid, right? I was good at it. I used to do parties, birthday parties and stuff. And one thing I learned about that is how easy it is to fool people. Mm-hmm. And mislead people, and right in front of their eyes, it's well, I saw it with my eyes. No, but what you didn't see is I did this or I did that, and I misled you somehow, right? And that's what I see when I look at this virology stuff. It's all so misleading. Mm-hmm. For example, there's something called the central dogma, and it's based upon how DNA and RNA is used to replicate cells, right? Living organisms. So any living organism that has DNA and RNA, it replicates. Mm-hmm. And it's so consistent, we call that a dogma. Except for one problem. Yeah, it happens in people, it happens in animals, it happens in plants, it happens in microorganisms, all living things. But when you get down to viruses, you find that RNA, again, the part of the gene in there, except that it's not used to replicate the virus. The evidence that's coming out now, and I spent the past year reviewing all the, not just the historical evidence, the latest evidence, right? And this is a problem with a lot of virologists. They're not up to date on the latest evidence, especially the professors who are just, they've got their lectures and it's the same old central dogma stuff. The latest evidence is suggesting, and I'm also saying it's still up to debate, but let's talk about the evidence that the viruses are waste products of cells. They're the genetic waste products of cells. They don't replicate. They can't replicate. They can't spread, right? Because the genes in them are not their genes. They're the genes that the messenger RNA used to replicate the proteins for that cell. And then the messenger RNA was no longer used, was fragmented and packed into an exosome. And that's what we call the virus. Okay. It's the waste product. And that goes into the virome, you mentioned it.
Yes.
Right. Yeah, and that's part of the immune system that transports that all out. So when we look inside these viruses, we say, oh, look, there's a gene, it must replicate, because everything that has genes in them replicates, right? I can understand why you would make that mistake.
Yes.
But it's wrong.
Okay.
And the proof of concept is, well, look, we took a petri dish and we put some living cells in it and we put some viruses on it. And then suddenly the viruses all disappeared. But then all these new little viruses started coming out. That proves it was replicating. Well, wait a minute. See, this is one of those magic tricks again, right? That viruses, first of all, if they're waste products, they're made of nutrients. Mm-hmm. They're made of nucleic acids, which are made of phosphorus and dextrose. Mm-hmm. And then there's the lipids and the proteins. There's practically everything a cell needs to recycle as nutrients. You put that on the cells and they break it down into nutrients, especially in a petri dish, right? And then what happens is, over time, the cells start excreting their waste products. the messenger RNA. And these are so-called the new viruses. So these are the progeny, right? The offspring. It's not the offspring, it's the waste product of the cells.
Yes.
You see what I mean about misleading people?
No,
Now,
for sure.
how would you prove that, or disprove or refute what the observation is there? You would have a control group. Another group, a petri dish with another bunch of cells. Instead of putting the viruses on, put something on that's similar to the virus, like that has proteins and nucleic acids, and put on nucleic acids and see what happens. You'd get the same effect. That's what my hypothesis is, right? But nobody's ever done that. At least I haven't seen it in the literature. Maybe they have. If you've seen this, let me know, right? Okay. And that's a quick and easy, quick and easy little, experiment anybody can do.
Yeah. And because
now we have the PCR test, so we can see right away, And so what we know is you're going to see the same reaction without putting the viruses in, right? That those nucleic acids will disappear because the lysosomes are breaking them down for recycling as nutrients to feed the cell. And then cell waste products are going to start coming out because as the cells replicate, they use up the messenger RNA in the ribosomes. Do you know this? Do you understand how this biology works?
Yeah, because the human virome doesn't necessarily cause harm. You got these latent and asymptomatic viruses. They're present in the body all the time, right?
I'll get to that in a second, but I'm talking about something inside the cell itself.
Okay, yes.
Okay. So what does messenger RNA do? Oh, it rewrites the code for one protein.
Okay.
One protein that the cell needs, because the cell constantly needs proteins to repair itself, right? Okay. Proteins wear out. People don't understand that. Right. In your body, your proteins are wearing out all day long, every day, and you need to eat a certain amount of proteins just to stay in what we call nitrogen balance, protein balance.
Yeah, so preach it. I'm a nutritionist. I'm always pushing protein on people.
But it's not a question of getting enough. It's a question of too much. It's a question of just getting enough to replace, unless you're trying to grow. And then you need a surplus. Yeah. But for adults like you and me right now, unless you're pregnant, right? You don't need excess protein. You need enough protein to replace the proteins that are constantly
correct. Yes.
Right. Well, that happens in your cell too. Okay. So your cell is always in need of proteins to keep on repairing itself. Yeah. Right. That's what the messenger RNA does. It takes a bit of DNA. It takes just enough code from the DNA to code for one of those proteins. Yeah. Right. So it doesn't do the whole genome. So then what?
So then it can change how you express certain processes in your body.
I'll fill you in. Okay. I'm just kind of teasing with you now. So once that transcription is made, the messenger RNA goes out of the nucleus. It goes into the ribosome. The ribosome is a part of the cell within the cytoplasm that reads that transcription. Okay. The messenger RNA is like a messenger. It literally delivers a message, the genetic code. That's all it does, right? Mm-hmm. And that genetic code is read like a ticker tape right through the ribosome. Mm-hmm. And as the tape is going through, the ribosome translates the code and says, "Oh, we need three of those codes equals one amino acid, a particular amino." It grabs amino acid and starts stitching together a protein, right? That comes out the other end of the ribosome. Pretty amazing, right? You should watch a YouTube video on this. So anyway, the messenger RNA goes all the way through and it's done. Okay. What happens to it? Sometimes it might be reused, but usually, well, I can't even say usually, we still are investigating this. It gets fragmented. It's a waste product. We don't want it anymore. Mm-hmm. Well, think about it. Maybe we don't need any more of those proteins. Maybe there's enough for now. So we don't want it to be recycled again and start creating more proteins when we don't actually need them. The cell's amazing. It knows how to manage all this stuff. So it breaks down that messenger RNA into 8 fragments for waste, and it packages them in a vesicle called an exosome, which then through exocytosis makes its way out of the cell, finally into the virome of the immune system of the body. That's it. So you can see that gene sequence inside the virome, inside that virus, has nothing to do with creating the virus. It's simply the waste products that were used by the ribosome of the cell to create a protein needed by the cell, right?
Gotcha.
So
Yeah,
this
gotcha.
contradicts the central dogma. Da-da.
Yeah.
That's it. You know what? I'm
Wow.
glad I talked to you about this. This is the first time I actually articulated it in this way. It's important.
But yeah, I slightly feel like I understand it more now than I ever have. Like that was a really well done explanation.
Okay,
because,
so if it's not replicating, right?
yeah,
How can it spread? It can't.
makes sense.
You could breathe in somebody's virome, but that's not going to mean that doesn't mean it's going to start replicating inside of you. Well, and then how come when we go into these nasal pharynges, we have these swabs and we see a viral load increased in there? Isn't that mean it's replicating? Well, there's another way to think of it. Maybe it's just accumulating. You see, what happens is in that virome, those viruses can pass out through your gastrointestinal tract, or they can pass out through your nasal mucosal system, which is part of your immune system, right? If you become ill for various reasons, and
that's where you flush everything out. So it's coming out that way.
Well, that's good. That's the compensation phase of being ill.
Okay.
But before we get to that, there's the toxic accumulation phase.
Okay.
Right? Now, these aren't really scientific terms. I'm now just describing to you my opinion of what's happening, all right? Based upon the evidence, though. I always base my opinions upon evidence.
Yeah.
I mean, as far as the opinions that I talk about in podcasts and stuff. I have all kinds of opinions. But anyway, so that's accumulating because it's not clearing through the nasal mucosal system the way it should be. It's getting backed up, right? So it's not replicating, it's just getting backed up. And why is it getting backed up? Because the nasal mucosal system is slowing down. It can't clear it. There's in the mucous membranes, there's the ciliary, the mucosal ciliary. What do you call that? I'm sorry, this is embarrassing. The cilia, that's it, sorry. I was thinking of the mucosal ciliary response. So the cilia are like these little brushes.
Yes, the little hairs,
and it pushes stuff out, right? And that includes viruses. There are certain things that you eat, for example, that can paralyze that. Sodium chloride.
Okay.
And this is proven. Not only is it proven to paralyze the cilia, the action of the ciliary response, but when it was tested with a virus, it allowed the accumulation of the virus because it wasn't being cleared anymore. I mean, it's right there. There it is, right there.
Perfect.
So the disease, whatever it is, let's say it's related to something we call nutritional immunology. This is a new science now. This is nutritional immunology. This is in my 4th paper, related to sodium toxicity. Okay, the sodium slows down, impairs the immune system, causes that backup response, right? So it's the toxicity that leads to the infection. What we call the infection is the viral load accumulation.
Yes.
Most people think of the infection as something that spreads, jumps into you, and hijacks the DNA of your cells and propagates. No, that's not what it is. This is what it is. This is an alternative hypothesis, all right? And this is based upon the evidence that I've accumulated.
So it's like the viral load bucket, and actually your toxic load bucket. How much have you accumulated of toxins, whether in your environment, in your toxic lifestyle, in your toxic stress load, the glyphosate exposure, the lack of sleep, the overtraining,
the overeating. Guess what? We eat 40% more salt in the wintertime.
Yeah.
Now, put that together as far as the seasonality of these infections.
Yeah, there you go.
Duh. Why do we eat 40% more? Well, we just eat more in the wintertime, and we eat heavier foods, right? And we sweat out less of the salt in the winter.
It's cold.
And that's enough to make a difference, right? All this is in my 4th article that is under peer review right now. I've had some good peer reviews. This is one of those articles, it's very polarizing. People either love this, it's like so eye-opening, or they absolutely hate it. Because it contradicts everything that they believe in, right?
Yeah, and when people are vested in a belief, it's hard to go the other way. But we should, absolutely accept that we can be wrong and move forward with the correct information.
See, you have the perfect balance between open-mindedness and skepticism, right? You don't want to be only open-minded and just go with everything.
I love everything. Yeah.
On the other hand, you don't want to be so close-minded and just be so narrow that you can't accept anything new.
Sure.
You got to have the perfect combination of both of them, right?
Yeah.
And so I could see what reviewers are, open-minded and critical and think, wow, I never thought of this before. And this is just a perspective article too. It's not proving anything. It's more to offer new insights for further study. That's the whole point.
Critical thinking.
So if this is true, if this is all evidence-based, then let's do some follow-up studies. Let's test it. Let's test a low sodium diet in long-term care facilities to see if it makes a difference in the infection rate in people where it is a really serious problem. Stuff like that, But other people look at this stuff and they go, oh, no, this violates everything that I ever learned about viruses. I'm not going there. They're closed-minded, I can't do anything about that. So I can just wait until I get the perfect combination of reviewers in the journal to finally get this thing published. But I think it'll get there. We'll see.
Okay. So anyway, I have a couple of minutes, but I want to do maybe some rapid fire with my last few questions. We'll just do quick responses to these last few questions and see if we can get just a few more nuggets from you, because this has been so good. Okay. So can you speak to any ingredients in there that are also a concern, whether it's the Pfizer, the Moderna, the Johnson & Johnson? Are there any ingredients that are like, you can quickly go, this one for sure concerns me?
They all do.
Okay.
I mean, I'll be honest with you. I haven't researched the safety factors of the vaccines. My research is on the efficacy.
Okay. I like that.
And also not the effectiveness. There's a difference between efficacy and effectiveness. Once the vaccines are out there, now you're no longer using experimental conditions. Now it's all observational studies. It's all effectiveness. And there are all kinds of problems with confounding factors. There's selection bias. Remember I talked about selection? I was talking about selection bias when you design a study to test for either severe infection or mild infection. You need different groups of people. Well, when the same thing applies when you're doing observation studies. Depends what groups of people you're looking at, If you get the wrong people, you're biasing the results. So you can't go by that. So getting back to your question, as far as the ingredients, you shouldn't be injecting anything into you, that's going to prevent a virus that you can have other ways of preventing.
We have an immune system for a reason,
So my idea is, if it's not even effective, why bother at all? Let's just cut it off cut the head off the snake, and you don't even have to be concerned about all those other issues. Don't get me wrong. We need to investigate all that other safety stuff too, right? Sure. But if it doesn't even work in the first place, come on.
Yeah. Okay. So the next one. So now let's say somebody has taken it who's watching this, and now they're like, can I detox off of the effects of this? What would you say to that? Are there ways to detox or help what it has done to the body, the effects that it's doing to the body that we're not even sure of?
Are you talking about now the vaccines, or are you talking about, what can you do to detox on the vaccines?
Yeah.
I have no idea. Again, you know what? That's interesting, because if I had done a safety study, I would have taken it into that direction. So I would have followed up exactly like what you said.
Okay.
But I don't know what you can do other than just, live a healthy lifestyle.
Yeah.
I mean, when people are ill and they want to get better, I tell them they have two choices. And I guess this would apply to your question. You can try to treat yourself or get treated, or you can try to recover your health.
Mm-hmm.
They're not the same thing. People normally would say, well, why? I want to do both. I want to get treated and I want to recover my health. And that's when I tell them, but you can't do both. And they say, why not? Say, because treating a disease usually means working against the body.
Right.
It usually means suppressing a symptom. I mean, the body, even when it's sick, is trying to do the best it can under those circumstances to balance out, to reestablish homeostasis. You're sneezing, you're coughing, you're getting that mucus out and all that other junk that's coming out. That's the vicarious elimination. That's the disease process that's going to leave you healthier in the end, So trying to suppress that is a treatment that works against the body. Isn't it better to recover your health by just, allowing the body to do what it needs to do, but more importantly, stop intoxicating your body. start lowering the lifestyle risk factors that put you at disease for all these problems. You know what they are.
Yep.
And one of the four main ones, what are they? Cigarette smoking, overweight, exercise, and nutrition, right? You know how many people practice all four of those? What is it, 5%? So I know it's under 10%.
Yeah. And getting outside, getting away from devices, because we're now more bombarded with electrical radiation from our devices more so than ever in human history. So like we have to step outside of this and get out into nature, because humans are made for nature.
But you know what? People are prevented or discouraged from taking responsibility for providing themselves with the factors of health because they think, oh, if anything goes wrong, I'll just go get treated. The doctors know what to do. Right? Sorry. That's a big, big, big problem. You know? And you know what? The people I find who are the most protective of their health are the ones who have been seriously ill and put themselves in the hands of the doctors, and they realized, whoa, they don't know what they're doing.
Yeah.
And they get that eureka moment when they realize, this is up to me. I got myself into this mess. I got to practice the healthy habits to make myself well, right?
Yeah.
Those are the people I find who are the most dedicated to this because they learn their lesson.
Yeah.
The people who haven't learned their lesson are the ones that are most at risk of really having catastrophic experience when they do so much damage to their body that it's too late to even recover their natural health, right?
Yeah. And I'm reminded of like the fact that there's been a lot of people that have been hurt by blood transfusions. And I think doctors maybe are now just realizing that the body does really well rebuilding blood. It just takes a longer amount of time.
And you have to know how to do it too. You know the food that builds your blood the most?
Beets are good. Dark colored.
Green vegetables. You know, chlorophyll is the same as hemoglobin, except the difference, hemoglobin has iron and chlorophyll has magnesium, right? It's the same structure.
So that's what I was thinking when you were saying, you know, we can have a really good innate immune system to take care of things. We just have to, like, in my opinion, give the body the proper conditions, and it's designed to heal. We just have to get out of the way, like,
and not put in those intoxicants.
Yeah.
And some of the things that we think are healthy, and vegetarians are actually one of the worst at this.
Yeah.
Do you know that vegetarians have a higher risk of stroke than non-vegetarians?
Yes.
Why is that? I was writing an article, a peer review, and one of the reviewers pointed that out to me. So I had to come up with an explanation. Well, you look at what they eat, and it's a high salt diet. I mean, Himalayan pink salt, sea salt, you know, Celtic salt, all these processed foods that are supposed to imitate animals, and you know, all the fake meat and stuff that's just loaded with. So, do you ever have one of those Impossible burgers? I mean, it's so salty you can't even eat the thing. You know. And so there's an example, right? Wait, people think they're doing something healthy and it's not even healthy. So you have to educate yourself, right? And don't get me wrong. Don't just take what I'm saying on face value. Investigate it, right? Debate it. I mean, the last thing you should do is say, whoa, Dr. Brown said this. So no, don't hold me accountable in that way. And all I'm saying is, investigate the stuff for yourself, right?
Yeah. And
find out what's healthy. You know, there's a section and that I'm hopefully developing in Trial Science News. I'm dedicating to, we'll see how that goes, to lifestyle medicine.
Yeah.
Which is all about the healthy factors, the lifestyle factors. That's it. That's where health is. Health comes from healthful living.
Yeah.
Right? A healthy lifestyle.
Yeah.
But we need education in that. We need information. We need research in that. We need information. We need communication. We need to design our whole economy on that kind of thing. Right now, we're designed our whole economy on sickness and the things that make us sick.
Yeah.
You know, I mean, people will get what they want. When people see that there's a better way, then they can start making those choices. And then the supply and demand will start shifting for those types of healthier things.
Well, Dr. Brown, this has Dr. Brown, this has been awesome, and you are just a wealth of. I could talk to you all day if I had enough time. So
go out for a walk. That would be the best way.
That's what I want us both to like. We can go from here. Everybody watching, we can go from here, go outside, get some fresh air, get some vitamin D, but also do your homework, do your research, critically think about things, make informed decisions, not just decisions based on the media and what you're being told. And if they want to find out more about you and your stuff, how can they get in touch with you, or where can they find you, Dr. Brown?
Well, you can find my work, and my email address is on all my articles. Just go to Google Scholar and put in my name, Ronald B. Brown.
Yeah. And I'll put a link in the show notes too.
Yeah.
So I so appreciate your time. I can't wait for your other stuff to read as well. I enjoy
I can't wait either. Boy, there's a lot of stuff that we got to get out there. We have to fix what's going on right now, and we got to make sure it doesn't happen again.
Good. Yeah. And I appreciate people like you out there doing the footwork and making it happen. So
you are important.
Keep doing what you're doing.
What I do is meaningless, and unless I have people like you to disseminate this information to the public.
Well, I'll keep doing it. We'll have you on again.
Great. Can't wait.
All right. Well, I'll let you go for now, and thank you so much, and we'll talk soon.
Okay. Bye.
Okay. Dry skin, hair falling out, sleep issues, low energy, constipation, digestive issues, even trouble swallowing, and feeling like your neck has a lot of tension. These can indicate a thyroid issue. And now, more than ever, I'm seeing subclinical thyroid issues in virtually every client I work with. So what's one easy way to amp up your thyroid and improve your health overall? Natural desiccated thyroid. And no, you don't have to go to your physician to get that. You can actually get natural desiccated thyroid from Ancestral Supplements. It's a company that I love and work with. I love their beef thyroid, beef liver supplement. Did you know that you can only make the T4 to T3 conversion into that active thyroid hormone if the liver's in check? So get your thyroid and your liver in check with Ancestral Supplements beef thyroid. Click the link in the description and use my code JODEL to save 10% on a great thyroid support that your body will love and it will thank you for in the way of getting rid of all of those symptoms I mentioned at the beginning of this.